I'm an R&D engineer at Kitware, and the name of my talk here is HistomicsTK Seamless Analytics for Biomedical Microscopy. So this project, HistomicsTK, is funded by a diverse group of participants and coded by even more people since it's open source. I work at Kitware. I work with a number of other engineers, and our primary collaborators on this effort have been at Emory University, although I definitely want to acknowledge some support of people at Beth Israel Deaconess Medical Center, Harvard University Medical School, and a few other places. And the work's been funded by the National Cancer Institute under their Informatics Technology and Cancer Research program. And it's an ongoing 5-year effort, and this is only year 1 of what we've accomplished. So, as I mentioned, I work at Kitware. For those of you who haven't heard of us, you probably may have heard of some of our software. Kitware is an open source software consulting company, and we work in a variety of domains. You may have used CMake if you're building Qt, KDE, a lot of C++ applications. We also have VTK for rendering, ITK for the analysis of medical imagery, ParaView for high-performance computing visualization, and we also do a lot of smaller projects, many of which I'll touch on in this talk. So the imaging modality we're dealing with here is whole slide imaging in microscopy, and this basically involves taking stained slices of tissue, oftentimes human, but many cases in animals, and digitizing the entire portion of the slide as opposed to just a region of interest. And this often results in images that are up to like 100,000 by 100,000 pixels across, and typically are required at 40x magnification. So just to show you, here is what is a fully zoomed out tissue block, and then you can get this level of detail where you're making out individual nuclei. You can tell which ones are undergoing mitosis, and you can see a lot of really interesting structures. And it primarily right now is dealing with bright field microscopy. We do a little bit of fluorescence and immunohistochemistry, but it's not anything too exotic quite yet in what we're working on, but it certainly applies to any modality that involves imaging very large surfaces. Now, the state of the art in microscopy in clinical world is still actual physical glass slides and microscopes, and if you want to collaborate with multiple people, you all crowd around one of these big lens microscopes where you have multiple viewfinders for each user. But luckily, in research and in a little bit of education, whole slide imaging and digital techniques have started to catch on. And this, in many cases, is almost a completely automated process at every point after you end up with an actual glass slide. We have robotic imaging devices. We have automatic ingestion and curation algorithms. And what I'm going to talk about here is an attempt to improve the tools for workflows and the analysis and data mining of images within whole slide imaging for cancer research. So to talk about how these images are actually stored, just to give you a background, like I said, the images are often about 100K squared, 2 gigabytes maybe compressed, which gives us terapixel resolution. And unlike many other medical imaging fields that use, say, DICOM to store their format in a standardized data way, whole slide imaging, although it has a DICOM specification, as a matter of fact, none of the vendors that are out there really support it or use it. So we get images from a variety of different formats. Most are actually sort of derived from a multi-resolution TIFF, but they all have enough dialects and strangeness that even just data, ingestion is a significant problem. But the way we'd like images to be stored and the way that we convert images to be stored once we get a hold of them is this idea of a multi-resolution TIFF, where you have a base layer of tiles. Each tile is like 256 squared on average, and then you recursively downsample it by a factor of 2 to produce smaller and smaller images that can be zoomed out. And this is the same technology that Google Maps uses. It's how really any large 2D surface imagery is displayed if you want to stream it. And actually adding this pyramid structure, adding this recursive downsampling structure only adds 33% to the file size. So it's something pretty nice. Metadata is, of course, a problem, as always, if we're not using DICOM. And that's something that we would like to address with future efforts, but at the moment we're focused more on the analysis of the images and less about curation of the metadata. So the need here is that cancer researchers, get, a large variety of slides. One of the resources we're using here is the Cancer Genome Atlas, TCGA. It has, 16 terabytes of images, and they need a way to manage it, and they need a way to easily access it on a variety of locations and share their results, and they need a way to run their algorithms on it in a way that's actually usable by both software people who can execute bash scripts, who can SSH all over the place, and more clinically focused people who really just need a GUI put in front of them and access to the algorithms, who can then actually make sense of what what's going on clinically in these images. There are a lot of solutions out there, and I'm sure you may, if you're familiar with this field, you may know some of them, but we haven't found any that is actually open source and free to use. And academic licensed, unfortunately, makes it difficult for people like me who work in industry or people who want to ultimately translate these results to be usable in hospitals. And we also don't find anything that's web-based or really supports analytics properly. And we feel that web-based is critical. We've had a lot of success using web-based platforms for pathology in education, and it's so much easier just to deploy things when you can, tell people to refresh their page as opposed to dealing with installers, network bandwidth, et cetera, et cetera. So what we decided to do here is use the Girder data management platform as a basis for building our own analytics framework for histology images, or microscopy images more generally. It's based on the Girder data management platform, which is a project that we've developed here at Kitware. It's quite similar to Midas, which is also a data management system, which many of you may have heard of. But we really like Girder because it has excellent software process. It's documented. It's got a RESTful API. It's tested. We have multiple clients. It's all Python based on the server. And it has a number of features in terms of user management, group management, provenance, tracking, task execution, the sort of things that, these are solved problems. No one wants to, write their own user management system just to ultimately get access to their images. So most of what we do here is based on top of the Girder data management platform. And you're welcome to check it out or talk to us later if you'd like to use that for your own data of any format. So these are just some illustrations here of what Girder can do. We have access control, which is important because in many cases, you may be dealing with images that aren't, maybe they're inside a firewall. If they have PHI, if they do have PHI, they're probably not outside the firewall, but you may not want to share them with everyone quite yet. We have a RESTful API is shown over here. It's documented and interactively usable. And it's something a lot of our work is based off here at Kitware. We also are using, as another thing I'd like to mention, GOJS. If you're familiar with, the geospatial domain, there's a number of viewers out there for large 2D surface imagery, Open Layers, Open Sea Dragon. We have GOJS here at Kitware because we found that the existing ones didn't provide a good mix of low-level access to WebGL primitives that's necessary if you want to do really high performance. But also good software process that were properly tested and documented and extensible. So those are the building blocks, and then and those already existed based on other efforts that we've done, and we wanted to put together a solution here for analytics. So there are two directions that really the analytics world takes. One is optimizing for single images. And that's sort of interactive viewing where you pull up an image, you choose some parameters, you want to minimize the turnaround time between the time you hit execute, look at what your algorithm does, get results, and then iterate and see if you can explore, say, the segmentation of nuclei or the extraction of certain features. Versus batch processing of images where you want to optimize on maximizing throughput. So you have, say, 100 to 5,000 images, and you want to execute an algorithm across all of them simultaneously, and then extract the results and do some aggregation of the statistics and see if you can look for patterns. And in many cases, this may then turn into a machine learning problem afterwards. In our case, we're focused more around viewing individual images because this is what ultimately allows us to work most closely with clinicians who they only understand individual images. If you give them aggregate statistics about extracted, features, you can draw conclusions, but you can't actually see the clinical correlation between this physical feature in an image and this, this number of an algorithm. But everything we're doing here absolutely applies to broader batch processing and map-reduced data sets where you may actually have, correlations between images that need to be considered when you're doing your analysis. And we also, of course, need to be able to have reproducible results. And it's great if you have something open source, but if it depends on packages which are no longer, supported or no longer even available, if you can't, if a Git repository disappears, if, important C library or a certain version of an operating system disappears, 5 years later, if someone wants to reproduce your work and they can't, that's a big problem for reproducible research, for science in general. And so you'll see we use Docker to solve a lot of those problems. So HistomicsTK is what we call our effort. Histomics is, of course, histology, microscopic tissue, plus omics as in proteinomics, genomics, et cetera, because we're really focused around the analysis of large numbers of these images, ultimately. So everything we have here is open source. It's Apache licensed. You're free to use it and fork it and do whatever you'd like with it. But of course, come and talk to us if you find it useful. It's tested, it's documented. We have a reasonable deployment process right now that's being improved. At some point, I'd like you to be able to just say git clone vagrant up, and you have a working environment. So to talk about the structure of how HistomicsTK actually operates, there's sort of 5 steps within it here. First is the actual domain-specific algorithms related to histology. We use, of course, Python wrapped ITK, simple ITK, NumPy, and scikit-image in our current efforts. But in theory, anything that fits inside a Docker container that you can, define input and output on, would be supported by HistomicsTK. There's a containerization step where we basically specify what the inputs, what the outputs are, and we deploy it to Docker Hub and leave it there in perpetuity. So if someone in 5 years wants to come back and run against our same algorithms, they can. And those algorithms can be executed directly, or they can be loaded into Girder and exposed via the web, and then run interactively by a user. And then once a user basically selects the parameters that they want, the execution is kicked off. Currently, it's using Celery. It can run remotely or on the same machine. But we'd like to move to like a larger, something that is either based on Hadoop or Apache Spark or something else if we want to do true batch processing with data locality and all those nice things. And then the final, component here is to look at the results and either render them on the screen or output them for future analysis, or even pipeline together multiple stages of an algorithm to, maybe you're going to run, a feature extraction on individual images, and you have another algorithm which takes all these aggregated data and pulls it together. So to talk about the first step here, again, our development right now is focused on Python just because we find it much easier. But ultimately, anything that you can, write in a Docker container, C, C++, if you could get MATLAB running inside a Docker container, we could make it work. You would also define via what's called the slicer execution model. If those of you have heard of 3D Slicer, it's a primarily radiological image viewer or image analysis platform. And that has basically what's called basically an execution model that defines inputs, outputs, execution, state, things like that. And this is a well-documented XML specification. And in this case, what you have to do is just define what the inputs are in terms of scalar values, enumerated values, strings, images, files, et cetera, to your algorithm are. Define the outputs, and then list your algorithms in a JSON file. Put a little bit of boilerplate Docker file text together. And then you basically have a running Docker container that we can deploy and host in perpetuity. But of course, that's not all. And although we do have like a really, somewhat comprehensive baseline of algorithms we've already implemented and are already available in our Docker container. And of course, we provide, via Conda, NumPy, scikit-image, Python wrapped ITK, simple ITK. All these image processing libraries are available to you in this Docker container. Again, people are welcome to, and we're exploring efforts right now to, take arbitrary algorithms in a Docker container and deploy them. But next, oh, I'm sorry, I skipped a slide there. Yep. So this is a list of algorithms we have implemented so far and technologies that we're using at the moment. So to go back to this, so this already I think is a pretty good step forward in how to do reproducible science and how to do reproducible algorithm development, to put things in a Docker container, have sort of a boilerplate to do I/O for you, and then really focus on the core details of the algorithm. And they can of course be executed directly. But another part of what we've done is we've used this specification of algorithm input and outputs via the slicer execution model to automatically generate web GUI components. So if you, as you see on the left here, if you define some parameters of your algorithm, it accepts, some string enumeration, it accepts some, some arbitrary, integer, we will automatically generate HTML GUI components that allow a user to select those values. So these are, we have a listing of a dozen or so different parameter types, and we have GUIs that automatically are generated based on that. And I can show that off in a little bit if this live demo works. So once your algorithms are loaded and available via the web GUI, a user can load an image from Girder. They can choose parameters, and they can kick off execution. And the actual execution, at the moment, we have an extension to Girder called Girder Worker that runs on top of Celery that actually manages execution. It handles, scheduling of jobs. It handles, serializing of results or serializing of I/O more generally. And right now we have active efforts in order to integrate this with Apache Spark and have a much more scalable system where you would have, Spark, if you're not familiar with it, will basically manage both your data via Hadoop file system and your algorithm execution. So to ensure that if you have, say, a parallel algorithm and data that's distributed across a distributed file system, the parts of the nodes where the data or the algorithm is being executed are right next to the nodes where the data is being stored. And we also are exploring right now MapReduce on the tile level as opposed to just the image level. So you could take a single image, you could split it up into individual tiles, and you could execute some algorithm on each subtile in parallel. But of course, you do run into cases where, like, what happens at the edge of two tiles? Maybe if you're segmenting something, which tile gets to claim the segmentation? And those are something which we're working on right now to abstract away and make available to algorithm developers. And then finally, when it comes to outputs, we always store the provenance of every job, the time it was executed, the parameters it was executed against, the version of the Docker container it was executed against. So in theory, if you have a database that has results, you could come back and in 5 years you could run the exact same deterministic calculation and get the same result. For vector outputs, we render them using GOJS and we store them in JSON. We considered using GeoJSON proper, but it lacks support for a number of different primitives like circles and rectangles that have parallel sides and things like that, and also provides no schema for style information other than just an unstructured properties object. And we also deal with raster outputs like heat maps, color normalizations, etc. And we output those as these multi-resolution TIFFs that I described before. And of course, because we're using Girder, all of our outputs are access controlled, and you could share the results with one person and not another one who is a user in the system. So that pretty much wraps up the talk. I want to leave a little bit of time for questions. I also want to mention future efforts here. So like I said, we're working on Spark integration. We're also working on generalizing this framework to be used beyond just histology. In theory, there's any sort of image analysis could use this model of putting your algorithms in a Docker container, automatically generating web GUI components to run them, and then abstracting away the parallelization process so that users can focus on either the algorithms or the interpretation of results and not the pipeline in between. So these are the people I'd like to thank again. And are there any questions? Yes.
Is there a publicly based course?
Yes, there is. In fact, I wanted to also have time for a live demo. So let's see if we can do this. So because this is in collaboration with Emory University, we have a public instance right now at girder.neuro.emory.edu. We have the entire TCGA, Cancer Genome Atlas, which is 16 terabytes, like I mentioned, and contains all different samples from several dozen different cancer types. And we've ingested that. We've got full metadata on everything TCGA provides, and we've got an interactive viewer here if you actually want to, zoom in and look around these images. And then HistomicsTK itself is also running on girder.neuro.emory.edu. Because our computational resources are a little bit limited right now, you actually do have to be logged in order to kick off analysis jobs. But you'll see here I have, the parameters queued up where I have, certain things for, nuclei detection set up. And I can actually toggle on and off. I don't know if you can really see this very well, but you can toggle on and off the overlays that are generated by this process. And if I were to kick off an analysis, it would probably take 60 seconds or 30 seconds or something to run. But I don't want to risk it right now. But I did generate these results earlier today. So, any other questions? Yes.
This is really great. Can you say a little bit more about how you can save the results? Like, can I share it in a Jupyter notebook?
Sure. So all of these are saved back to Girder, and Girder is backed by MongoDB. But one of the great things Girder provides is and I'll show you here, is a RESTful interface to all of its data. And we also have a Python client that provides convenience wrappers around all these methods as well. And so it's absolutely something that our collaborators do, that I do, is to use Jupyter Notebooks as an interface to this data. So, ultimately, you don't even have to use our Python client. You can really just use the requests library or any, HTTP library to get this data, pull it in and out of Girder, do results on it, or excuse me, do processing on it locally and push it back up. Alternatively, if you wanted to work directly on the back end, like I mentioned, HistomicsTK is capable of being run directly without, from Docker without having all this web interface. So if you wanted to write your own, Jupyter Notebook, script that, managed your images yourself and just invoked one of these containers to run an algorithm, you certainly could as well. And further integrations between GOJS and Jupyter Notebook are actually something we're going to be doing in the next couple months. And I'd really like to see an interface where you have, a real-time updated display of an image like this, where you're, on the left side you have some code, you execute it, and on the right side you immediately get updated results right in the notebook, as opposed to having to go and fetch them and pull them down from the web.
What's the Python called?
It's just called Girder Python Client, and if you go to github.com/girder/girder, it's fully documented there. We have examples. It also has a CLI, which actually is wrapped around the Python client. So in theory, you can do batch upload and download right from a bash, command line.
And you can pip install Girder-client.
Yep. Actually, that's probably an even better way to get it if you just want to run it. Pip install Girder_client.
We probably have time for one more question.
Sure.
The next speaker just start setting up while we take that question. Do we have one more?
Can you talk about all these algorithms and can you run them from the API, the web API?
Yes. So the web API allows you to kick them off directly. You would need to pass in the inputs as a slicer execution model, formatted XML file. But you can actually use the RESTful API to run the outputs, the inputs, although most of our use case right now is focused around if you want to do batch processing, you'd run the Docker containers directly.
Git install girder.client.
Girder_client. Yes. Oh, yeah, might be dash. All right.
Thank the speaker again.
Yep. Thank you.